Today we're going to be seeing what are the best approaches to your own experiments design. There's not going to be that much practice in the computer today because it's mostly mental work. It's mostly about applying the things you already know, understanding the things you don't know, and trying to use all those things, in order to find the best path, the best approach to tackle your research objectives. That's basically what we're going to do today. No programming involved, no typing, so I need your minds focused. Also, I asked you yesterday, most of you, I guess, got to listen, if you could prepare your small presentation, because we're going to have some spare time during the day. And if we can use whatever we talk about today in order to help you focus or better find new ways to approach your research objective, your personal research objective, and also understand how this course will help you reach those objectives. All right, so welcome back. Do you have any questions at this point? Whatever it is regarding previous days, regarding things that were not clear that you'd like to retouch later today, or data that is not available, or data that you would like to have available. Whatever it is, this would be a good time, or at the end of the day as well. There it is. Okay. So, first thing, a very quick disclaimer. I will not pretend to know all the methods that exist, what are the limitations, or all the pros and cons of each and every single kind of experiment design that you have in mind. Each researcher will have its own his or her own circumstances, and they will decide how to tackle each step of your research, taking into account your own capabilities, your own skills, your own infrastructure availability, your own budget. So this is the first thing we need to know. So we're going to touch some overall general methodologies that you're probably thinking when you talk about bioinformatics. But bioinformatics goes way beyond what I'm going to show here. More often than not, you probably think of bioinformatics as dealing with sequences. You get sequences from a sequencer provider, and we bioinformaticians, more often than not, we get just ask a single question. What can you get from? I'll give you just 1 million reads, 1 million sequences from 64 different, or 10 million reads from 64 different varieties or species or samples, etc. Please, can you find something there? That's usually what we find. So, from our point of view, this is a problem in experiment design. Why? Because we didn't think, or whoever ordered the sequencing didn't think beforehand why they were sequencing whatever they were getting. That's the main thing. It's like, if you're going to sequence something, please have in mind, will this sequencing actually answer what I'm asking? And for that, you need to understand what are the limitations of the sequencing technology, what are the limitations of our analysis capabilities, what are the limitations of the hardware we will have to analyze the data you are offering. So all those limitations need to be taken into account. And it takes a lot of time, a lot of experience to know how far those limitations will affect the results. This is going to be a quick outline of the presentation today. First, we're going to talk about different kinds of research, the two main branches that we have today. The traditional hypothesis-driven research, we are all familiar with it. We've been taught our entire life how it works, but more often than not, we don't really apply it in our science. We would like to, but we haven't been taught how to properly use it, especially when we talk about big data, a lot of information. Then we'll go and see what is data-driven research. This could be a little bit of a subjective opinion, but basically we'll see how the huge amount of data have affected the way we are tackling research at this point, and how we can improve our uses of massive amounts of data in order to tackle very precise, concise objectives. Then we're going to talk about how you formulated objectives. I'm going to give you a few examples of objectives, of overall objectives that look nice, sound nice, but overall are kind of useless. Why? because you don't have a clear idea of what you're trying to tackle. Then we're going to talk about how background research to actually do your experiment design is useful. How this background research, all the literature research that you do for your thesis, for your papers, for everything you do, it's absolutely necessary. If you don't know what's already available, you're going to be repeating a lot of research that has already been done. If you don't research, if you don't understand what are the tools that you're proposing to use through background research, you will be using methodologies that will probably not answer the questions that you want. Finally, we'll reach how to select the methods. More often than not, this will include trying to talk to somebody who is specialized in each of these methods. You can have an overall understanding of how each method works because you've done your research, but the devil is in the details. All those small steps that you, for example, I guess a lot of you have already working in the lab, right? You've done in molecular biology labs or you work in the field. And you know that when you write a paper and when you read a paper, it all looks like everything worked out in the first go. Like, how we did amplification in the first go and how we did the genotyping in the first go and nothing went wrong. But when you do the actual things on the field or when you do the actual work in the lab, and it's the same with data with bioinformatics, when you do the actual programs, you have a ton of problems. A ton of problems that are usually not reported by anybody. Oh, I put caps here and I didn't put caps in the next sentence. So, oh, I forgot to add tag in this experiment and nothing amplified and I just got crazy. Oh, I got some contamination in my samples and I didn't realize it until it was too late. So I have to resample. All these kind of things that nobody reports, they happen all the time. So these are the details. How attention to the details on each of these steps has to be monitored or at least has to be advised by somebody who has a lot of experience in each of these steps. We cannot pretend to know every single thing about every single method that we're going to use in our research, but we will learn. And finally, identification of those limitations and caveats. These people who are specialized in each of these methods and ourselves who are specialized in what we're trying to find in our research, that is exactly what it's going to tell us what are the limits of what I'm going to conclude. If I know, for example, that I'm using a machine that has only a certain resolution, I cannot claim in my paper that I found something better than that resolution. If I know that I only have in my sequencing, I only have certain depth to my sequencing, I cannot claim in my paper, just using basic statistics, that I have sequenced 100% of what I wanted, because sequencing works like that. It has its limitations. The same way that if I find orthologous genes between different genomes, then I have to say, there are limitations because I'm using a tetraploid. Some of these are probably not orthogonal, it's a paralogs, or, well, there's a lot of different limitations that specialists will need to help us. We cannot pretend to know it all, and we cannot pretend to know it all in our research papers or in our reports. So, this is now the outline, so let's start with the basics. We all know this hypothesis-driven science. We have an observation, we observe something that we think it's interesting, we ask ourselves questions. Usually the questions are very broad, like, why do you think this will happen, which is very broad. What happens if I do this, which is a lot more specific. When we start asking those questions, the first thing we need to do is try to find what has people asked before. Has anybody ever asked the same questions I have? If not on this species, has anybody ever asked the same question I'm asking in another species? If they have, how did they tackle it? What methods did they use? Would those methods work in my own research, or they wouldn't work? Why they wouldn't work? All those things come from the background research, and that's why it's so important. Let's assume that you want to find diversity in a population. This is just a very typical example. Diversity in a population of sharks in the southern Pacific Ocean. Let's just assume that, all right? And you see that a lot of people have done this kind of work. They've done genetic diversity using microsatellites. They've done genetic diversity using SNPs. They've done genetic diversity using AFLPs, rapids, etc. All kind of markers. And each of those markers have their own way of measuring diversity in the population. They have their own resolution, etc. And you say, well, okay, what's available for shark? Does shark have, or for my shark, does it have a reference genome that I can use to quickly find SNPs? If the answer is yes, then you know, hey, maybe I can go for a SNP. If the answer is no, then, well, I still want to go for SNPs, but I would have to develop some genomic resources first, like a reference genome, in order to get better SNPs. Or maybe I can go for a reference-free SNP discovery pipeline. Yeah, there are many options. They say, no, let's drop it. I don't want SNPs anymore. Let's do something more classical. I have already some microsatellites from another shark. Yeah, let's use those microsatellites and see how they work on my new species of shark that nobody has ever researched before. And then you do some filters and say, oh yeah, it kind of works, it kind of doesn't. So the first thing you need to do after you do that background research is understand what is already available, what you can use to further your knowledge about whatever you want to find out. Then once you have a clear idea of what is available, you will also have an idea of where are the gaps, what things you have thought about that nobody else has. Yeah, it's, for example, you say, all right, I want to know the diversity of this shark. And I already know that, okay, there are different libraries, etc. Oh, but nobody has actually produced a FOSMIT library for this shark. Nobody has produced a BAC library for this shark. Nobody has tried long-read sequencing for this shark. So you start saying, oh, there are gaps in knowledge. There are gaps in genomics resources. Now the next thing is construct a hypothesis. You already have as much background research as you can. You have to limit yourself because you can do background research for years and years and never actually finish reading all the literature. But then you start formulating your own hypothesis. Now, what does this imply? What is formulating a hypothesis? If anybody can help me here. What does formulating a hypothesis means for your research? Does anybody want to share a little bit? I'll share an example from my personal experience. A few years back, I was trying to isolate a resistance gene in potato. It was a single locus monogenic gene. We kind of knew where it was in the genome thanks to genetic mapping. And we wanted to know if we could isolate the sequence. Now, why would I do that? What would be my hypothesis of working? My hypothesis of working was, at that point, after getting the genetic map and getting linked markers, was, all right, my gene is genetically linked to these markers. It's very close to it because I know the centimorgan distance. Can you hear me? Yep. Yeah. So if the gene is, as it says, linked to these markers, if I sequence parts of the genome around that marker, I just might catch it. I don't know how far away in basis it is, but I just might catch it. So I say, all right, that's exactly what I want. That is exactly my objective. I want to isolate that gene. How do I go about it? Then we start testing with experiments. All right, what experiments can I do? There are 1,000 experiments. I can do genome walking. I can do map-based cloning. I can do, well, there are several things. I have to choose one. What does it depend on? It depends on my budget. Depends if I have the plants, if I have the DNA, if I have the resources to actually get the sequencing done, etc. So what I did is, since I was assuming that my gene was close to these markers, what I did is we produced a BAC library. We selected some BACs based on the markers because we had the markers primers and we could select those BACs. Yeah, and then it was a matter of, okay, I've got BACs, but I don't have a sequence. I need to get a sequence to actually see if there are genes in these regions that are close to the markers. Yeah, so we needed to do sequencing. And after doing the sequencing, we could get just a lot of reads, small reads. So now comes the step, we needed to assemble them. Assemble it to actually make it a single consensus sequence for each of the BACs and see if the BACs are contiguous. And there are a lot of tests that needed to be done. And after that, after we had a single sequence for the entire locus, they said, all right, so are there even genes here? My hypothesis was that there must be genes. Yeah, so if I run a gene detector software, I should find genes. That's my test. Or if I find RNA data or cDNA data that aligns to this region, then that means that this region probably has coding sequences, genes that are actively transcribed. Yeah, so for example, these are two extra hypotheses that come afterwards that will help me tackle the next question, which was, are there genes in this locus? Yeah. Then I do some tests. I do the cDNA. I sequence that with Sanger. I align them to my locus. I find a few reads that align. When I do a blast search, I also find that some known proteins and genes align to those same regions. I say, okay, this is potentially a gene. Can I find a better structure of this gene? Yes, I can. And we run some gene models. We produce some specific gene models with exon, introns, start coding sequences, encoding sequences, etc. We found a few candidates. Say, all right, cool. We've got a few candidates. Now we go to the main question. Is that my hypothesis was that at least one of these candidates was conferring resistance to a specific virus. The only way I could think at that moment is, well, the only two ways I could think of at that moment was, first, I can clone each of these genes separately into a susceptible variety, and if the susceptible variety becomes resistant, then that would mean that, or I could do it the other way around. Okay, I can take this resistant variety, and I can try to knock down each of these genes. And if, when I knock down one of these genes, the variety loses the resistant phenotype, then I could assume that, yes, that was a gene that was confirmed resistant. If you notice, I've done several iterations of hypothesis building, experiment testing, then checking if the results coincide with what I was expecting or if it didn't. Yeah, but everything was, in this case, kind of driven by hypothesis. However, science doesn't always work like that, right? As we were saying before, every time we test with an experiment, and this is the nice part about constructing a hypothesis, that the hypothesis needs to be testable, we don't know if the procedure will work. Maybe we've seen it work in a different plant or in a different project, but for our plant, unfortunately, it doesn't work. A typical example, at least from my experience in potato, it was when you work with a sweet potato, it grows everywhere. You have no problem making it grow in the field. But potato doesn't really, it's really spoiled about what it needs to grow in the field. It requires a very specific kind of soil, it requires a very specific environment, and each variety of potato requires a very specific environment. Like, and I don't like this soil, sorry. This soil that worked for your white potatoes in the coast, they don't work for me. I'm a native potato, and I live above 3,000 meters above sea level. So I don't like these conditions that you're giving me. I need better conditions. I need more specific conditions. It's the same with bacteria. For those of you who work in bacteria, yeah, some bacteria like a generic nutrient growth. Another bacteria, they like, no, I want vitamin A, and I also like some protein included in my broth, and then also like, so they are very picky about what they need to grow. It's the same for everything. So these procedures that we've seen work in other papers, they probably, or they may not work in our own research. So we, more often than not, will need to try to tackle those issues. And that's going back to what I was talking about the experts in the field. What conditions can we tweak to make the procedure work? Or do we need to just overall scratch it and move on and try to find a different procedure that works for me to test with an experiment, to test my hypothesis? After we get the tests working, yeah, we got data. We got data, yeah, but more often than not, people don't think beforehand, how am I going to analyze the data I get? This is a big issue because it goes back to what I was saying about people giving us sequence data, but since they don't know how they're going to tackle, how they're going to get the metrics they want or they need, yet they don't have a clear idea of how to analyze the data. And if you don't have a clear idea of how you're going to analyze the data that you are going to produce with your test, then you don't have an idea of how to test your hypothesis, how you're going to tackle your main objective. If you think, or maybe sometimes it's just that you haven't thought about it. It's just that you thought the data you were going to get, worked differently than the way it actually worked. So, so the procedure you selected and, and all the tests that you've run, in the end, when you finally get the data, and you can get tables, you can get sequences, you can get scores, you can get a lot of different data to analyze, you don't know how it actually helps you further your research or get closer to your final objective. So that's something you also need to be clear. And for this, usually what I mean is a statistical analysis. What kind of a statistical analysis of your data you're going to do, what kind of test, statistical test on the data you get you're going to use to actually prove or disprove your hypothesis. And this is something that you need to be very aware of. Well, then you see if the results align with your hypothesis or if they don't. If they align to your hypothesis, you say, okay, it seems like my hypothesis is more likely to be true than not, but it's not entirely true because each hypothesis has different elements to it. It's not only that one single test will completely prove or disprove my point. No, it will just support my point. If the results, in the same way, if the results don't align with what I was expecting, I can say that my hypothesis is not entirely correct. There are some things that I need to modify. It doesn't mean that it's absolutely wrong and that you have to scrap it. No, it just means that, it probably needs a little bit of better explanation, or I didn't get right this part of the hypothesis. It probably works this other way around. But doesn't mean that it's completely scrapped. No. It means that you need to keep reformulating your hypothesis until all the observable data that you have or that is available keeps aligning with your hypothesis. If some data that you get or that somebody else gets doesn't align with the hypothesis, that means the hypothesis has to be modified. Modified to actually explain the new data that is available. And now that you have modified the new hypothesis, you are able to go back and find new ways to test. Now, what about data-driven science? This is something we usually do when we are, this is actually something that we do more often than actually sitting down and thinking a hypothesis and thinking how to test a hypothesis and thinking if the hypothesis is clear or not. This is what we do. We do exploration. We observe something, we are curious about it, we don't have a really clear idea of where we're going, so we start exploring the data. We start trying to see, hey, is there any correlation between different parts of the data I'm analyzing? Yes, there is a correlation. Say, okay, so is there anything about this correlation that could explain something else, or is there anything interesting about this correlation? More often than not, we're pushing that direction because we need to produce some science. And science, we need to produce some publications. That's what's keeping our jobs, right? The fact that we need to produce some information that is relevant for the scientific community. So we say, all right, let's just produce data and try to see what happens. And this is basically the basis of data science. Data science goes back to the late 90s when the term data mining started being used. And what does data mining meant back then? Data mining meant that using statistical or novel statistical approaches, you would be able to look into already existing databases or data, like nucleotide sequences, maybe just all data from SSR, maybe some genotyping data from several species. Maybe you got some interaction networks that you had already found using some molecular biology experiments, and you had tables about the correlation between different parts of different molecules. But nobody knew actually how to handle it. And so we say, okay, let's try to use a very strict statistical framework that let us test different correlations. When we start doing that, we start discovering new things, new relationships. But here's a caveat. The fact that you find a relationship between different elements or different variables of your data doesn't mean that you have already found an explanation. It's just a correlation. It's like finding a marker linked to a gene. The fact that the marker is present kind of lets you know that, oh, the gene is present and probably the phenotype is present. Yeah, like the example I was given to you. The fact that I have a marker present let me thought that the gene was around, and the fact that the gene was around allowed me to think that, oh yeah, probably the phenotype will also be, the resistant phenotype will also come along with the gene. That was a hypothesis, but the fact that the marker is there didn't necessarily mean that the gene was there. And the fact that the gene was there didn't necessarily mean that the phenotype was going to be there. Because there are a lot of other variables that we need to tackle. But in this case, the fact that you find the relationship is still significant. Just because you don't know exactly what's the reason for the relationship, yeah, doesn't mean it's not significant. But this is also a limitation. The fact that since you don't know the reason of the relationship, it could be just a random relationship. It could be just an artifact of your statistical approach. It could be an artifact of bias in the sequence, in the data you're exploring. So doesn't necessarily mean anything. So basically, how this data science world works. Oh, sorry, I just jumped one step. So this was data mining, getting new information out of all data, a lot of all data. Yep. So once the statistical framework was established, they say, all right, why don't we use computers to actually speed up this discovery of relationships? Yeah, and that's where it started being called data science. When we started using computers and more elaborated algorithms to find those relationships, to find those relationships between the different parameters that we have already measured in all databases. And all of a sudden, all of these high throughput phenotyping and genotyping platforms started appearing. Yeah, and all of a sudden we had hundreds of thousands of sequences, genotypes, markers, a lot of phenotypes related to them, and people just stopped knowing what to do with that. Yeah, and that's when both data mining and the new high throughput sequencing methods and the computational capabilities to tackle those problems, those data mining problems, yeah, started being developed, that the data science actually started to take off. So more often than not, data science is mostly an exploratory data analysis. That means we're getting a bunch of data and we're just starting to see. We don't know exactly where this is going, but we're starting to see, hey, it could go this way, we could find this way. You find a bunch of correlations between your different variables in your data. Yeah, and say, okay, I want to tackle this correlation, I want to tackle this other correlation, I want to tackle this other correlation, because for you, you've already formulated in your mind an idea of, yes, this is significant. This correlation here is significant, and I think I can explain it. And that's where both worlds start combining. The fact that you've already explored the data and that you found some correlations, yeah, lets you focus on each of those correlations that you found, or maybe several correlations at the same time that you found, yeah, and formulate hypotheses that you can tackle later on by testable, better said, you can formulate testable hypotheses, something that you can measure, something that you can see, something that you can actually say, oh yes, I have obtained results that actually help me discover the truth or falsehood of what I'm saying in my hypothesis. So the exploratory data analysis works from here. First you have raw data collected. Yeah, the next step usually is clean up the data. Or how do you process the data is how you generate the tables, how you generate, do you remember how we got a bunch of sequences, a bunch of CDSs from the annotation file? Remember you had a bunch of, like 150 lines? And say, okay, I want to know how many CDSs there are, how many exons there are, etc. And we generate a table. But the raw data collected didn't actually give us that information. We had to process it, and we produced a table. But the table wasn't pretty. It was dirty, right? You had like one count of spaces or one count of NA. I said, yeah, that doesn't really tells me anything about the data. So Reinhardt the next day taught you how to clean up your data to make it not only look better, yeah, but also to just keep the relevant parts. Then you clean the data set. Then after that, the data scientists start using different models or algorithms, a statistical framework that actually lets you find correlations between, for example, the numbers and the notations, or the locations of the notations and the location of the genes. All kind of data that you can think, all kind of variables that you can think of that you can get from that information, yeah, you can try to correlate between each other. Sometimes it was something as simple as just discovering that there is a high correlation between the starting codon in a protein, ATG, yeah, and coding sequences. There was just a high correlation. We'll say, all right, yes, this actually looks like this is a thing in biology. For some reason, all proteins or all coding sequences start with ATG. Okay, once we have models, algorithms, we found correlations, we start trying to visualize, report, and communicate our results. This is important because if whatever we find, whatever correlation we find, even if we don't understand why, if we don't put it in a way that people not specialized in the area can visualize it, like some nice graph that show you, hey, this is red, this is green, green is good, red is bad, based on this threshold. So yeah, this means that we have found this good relationship between these two variables. Good. Yep. If we don't do that, people are not going to be able to take advantage of the data we are mining, of the data we are obtaining. The communication also needs to be clear. It needs to be very precise about what methods, what algorithms, what models you use to obtain those correlations. Just because other people who can also be experts on the field, yeah, may tell you, yeah, that doesn't make sense. Or they can tell you, hey, this actually makes a lot of sense with some other data I already had, but I hadn't published yet. So I see you're getting the same results. Now that you have a way of doing algorithms and you have some data produced, like some correlation data produced, you go back to reality, you collect more data, you repeat the whole thing, yeah, and you see if your models still apply to the new data you have collected. If your models do apply, then you keep using them. If they don't apply as well, you keep improving them. You keep improving these models and algorithms. This is all part of the exploratory data analysis. A lot of us do it. A lot of us do it without even thinking about it because it feels natural. It doesn't feel as natural to sit down and try to isolate each of the variables in your experiment and say, okay, I want to control this variable so that I can have an effect on this other variable, and I have to make sure that all other variables stay the same so that I can say that there is a direct causative correlation between the first variable I'm working with and the dependable variable that I'm trying to understand how it works. Yeah, so that's a tricky part. The tricky part of hypothesis-driven, and this is the nice part of data-driven. The fact that it feels natural to just, when you don't know what you're doing, or when you're not sure exactly what you're dealing with, you start exploring around, poking around, pushing things around to see, hey, yeah, this is moving just in this direction, until you actually make sense of the data. Now, this is actually a joke, yeah, where some data scientists discovered that there was a high correlation between the number of sales in the company and the fact that, the number of people that had just shaved their heads. So the company makes the decision that says, yes, let's all shave our heads because that's gonna raise the number of sales. This probably is an artifact of the data science approach. Yeah, the fact that this is a spurious correlation between sales and the number of shaved heads in your company. Yeah, but if not communicated properly, it can drive us to make wrong decisions. It can drive us to draw conclusions that are wrong. Yeah, you need to understand, and this is exactly going back to the hypothesis-driven part. Is that you need to understand, or at least propose a model of why you're seeing this relationship, yeah, and trying to make sense of if that actually could happen in the real world before actually accepting all the relationships that you see, yeah, as true. One of the most used, or the most recently developed methods that use this kind of big data, it's neural networks. I'm sure most of you have already heard about it, yeah, but few have a real good idea. Does anybody understand how a neural network works in its more raw sense? Basically, it's just as simple as this. Basically, it's like you have a lot of training data. That is, you know that if you have, let's say, for example, a marker A, yeah, and a phenotype B, You don't know how it works. You have no model, no idea. You have no hypothesis how this works. You just know that it looks like there is a correlation, and you say that if you have genotype A, you have phenotype A. If you have genotype B, you have phenotype B. And you have genotype C, you got phenotype C. And then what you start doing is that you train your neural network. The neural network will take the input, yeah, and also will take the information about what they are supposed to get. So if you tell the neural network, A is supposed to be translated into A. B is supposed to be translated into B. C is supposed to be translated into C. If you use very little data, the neural network, once it finishes its training, yeah, it's going to give you very bad results. Because you're going to give them actual real-world data, yeah, that it's not part of the training set. And it's going to say, oh, this is G. What do you expect G to form? Oh, I expect G to look like A. Why would you expect that? Oh, because in my data set, I've only seen A, B, C, and D. I have no idea what G is. So I'm guessing G is closer to A, and therefore, for my interview, G is closer to A, so therefore I will classify it as, yes, A is going to produce the A result. G is going to produce the A result. Because internally, I think it's closer, and I don't have any more data. So neural networks actually let you do very high fidelity predictions of what you're going to get if you feed them enough data. And by enough data, I mean hundreds of thousands of data points. That's basically what it does. It just finds a way to transform the original input data, your marker, your numbers, your database, yeah, and get you either a classification or a expected outcome, yeah, based on what it has already seen before. It tries to model this prediction, yeah, as if it was a learning brain. Yeah, I'm not going to talk about the hidden layers in neural networks because that's more of the algorithm. I just want you to understand how this works and how this relates so tightly with the data-driven research. The fact that we don't know what drives these relationships, we just know we've already proven they are true, but we don't know how the relationship works, but we still want to use that relationship in order to do predictions. These neural networks are the ones that help us do the predictions without actually knowing the background or the actual pathway from which you go from A, you don't know it, but you can still predict it. Because you have hundreds of thousands of data points, and you have a neural network that has been trained with those data points, yeah, so exhaustively that you can do predictions. The more data you feed it, the more accurate the predictions it will be. The more diverse the data you give it, the more accurate it will be. Yes. Basically, there are some metrics. How do you know it's enough? It's like when you start checking actual real-world results, yeah, and you get a correlation between what you would expect and what you expected and what you're getting from the neural network, yeah, close to like 92, 93%. You say, okay, this is enough. But very often they don't even reach that. They say, okay, 60% correlation, like the correlation between what I expected and what I'm getting. This is good enough. Yeah, I'll just leave it there. Yeah. Neural networks are currently used for pretty much everything. Pretty much everything. Just not too long ago, I was in a project. They were trying to, from RNA-seq data, they were trying to find out which of the transcripts were not actually coding sequences, but they were non-coding RNAs. And there were a bunch of tools, most of them depending on already existing databases. So you had to compare your transcripts to a database. And if the database had already discovered that, or had already annotated that, yes, this kind of transcript is a non-coding RNA, yeah, say, okay, you classify this as a non-coding RNA. But there was another approach that was using neural networks or deep learning, and they say, okay, if you give me a set of non-coding RNAs and a set of coding RNAs, train me, yeah, and then you give me your actual problem, your actual transcripts, and I'll do the prediction of whether they are or they aren't coding sequences, or if they are non-coding RNAs. We all are, yes, as the input, It's usually a one-to-one thing, usually. You try to do it as a one-to-one relationship between the input and the output. Let me see if I understand your question. So in a single neural network, yeah, the ideal thing is that your input is just one variable, your output is another variable. Yeah, that's for your training, yeah? But what it can get as an input, it depends on the kind of neural network that you're dealing with. Not all neural networks will handle the data the same way, or will actually be able to do the prediction in the same way. Some neural networks are specialized for receiving sequence data. Some neural networks are specialized for receiving quantitative data. Some neural networks are specialized for receiving interaction data from different proteins. So each kind of relationship will use its own kind of neural networks. And that's a very active field of research at this moment in computer science. The fact that you need to, trying to develop new neural networks, the objective is to decrease the number of data points used to train, yeah, and increase the sensibility and the accuracy of the predictions that it makes. So these are algorithms developed to exploit extensive data. Requires currently exhaustive training to increase prediction accuracy. Since they are trying to mimic the brain, the human brain, you probably have noticed that when you start teaching your kids anything, your kids anything, they take a little bit of time to discover that if you show them 2 or 3 chairs, they already know what all the chairs look like. They will start calling new things that they didn't see ever before. They say, oh yeah, that's a chair, that looks like a chair. And it does a very clear classification of that. Sometimes it makes a mistake, but it keeps training and learning, etc. Yeah? The human brain can work and make predictions using very few data points as training. Yeah? The neural networks, they need hundreds of thousands. So that's what I mean by they are trying to make it look or make it work more and more like human brains, so that they can get even less data, but they can still do similarly accurate prediction. And as I was telling you, it can discover previously a no relationship between variables, but it offers no explanation of those relationships. That's up to us. That's up to the scientists to try to create or design or think of a model that could explain those relationships. Now, to use hypothesis-driven research or to use data-driven research, that's up to you guys. They are both useful. Yeah, I personally prefer to, I like more the hypothesis-oriented way. Why? Because it lets you get clearer conclusions. Yeah, you can actually draw some really clear conclusions about what you're trying to achieve. Yeah, of what you're researching. Data exploration, it gives you information, but it doesn't give you. You can use that extra information to get or to inform your decision or your model making or your hypothesis. You can and you should, yeah. But just doing the exploration by itself, it doesn't really give you anything else about what your main objective is. If your main objective is, hey, I just want to develop a database for, I don't know, SNPs in sugarcane or in quinoa, yeah, I can. It's going to be useful. Somebody's going to use it, yeah. But is that what you wanted in your objectives in your research? Was it just to develop resources, or you were actually looking to tackle something bigger? And developing the resources was just the first step in that direction. That's something you need to ask yourself when you're trying to understand what you're researching. So, model research uses both approaches iteratively. You produce more data, yeah, because you have an objective in mind. You then formulate hypotheses based on the new data you develop. You do more specific tests that will test your hypothesis. You get more data from those tests, sometimes confirming what you're expecting, sometimes not confirming what you're expecting, and then you can get even more data from other colleagues using other plants in other species in other parts of the world with different conditions, but they are related to what you were doing. So then you use data mining to discover correlations between variables. Then hypothesis is used to try to explain those correlations and test them, and make predictions. Those predictions are exactly what we're going to test, because we're going to start pushing variables around based on our hypothesis. Then experimentation is used to see if those predictions were correct or not. That's going to disprove. Yeah, and this is also an important part. It's a lot of people have different opinions about what you're trying to do by experimentation. Personally, I like to say that I try every time to disprove myself, to try to prove that my hypothesis is wrong. That's what I like to do. If I fail to disprove it, I'll try to find a new way to disprove it. Another part of the hypothesis that could be wrong, and I want to show that it's wrong. I want to show myself that I'm wrong all the time. Yeah? Some people say, no, it's actually I'm trying to prove that I'm right. So, well, yeah, but that doesn't really inform me. We can learn a lot more from our failures than from our successes. Well, that's from my experience. So, further observations. Once the experimentation is finished, you can see if this new data actually violate your current accepted hypothesis or if it confirms it. Yeah, and you can go back, keep exploring, trying to find new correlations, and then keep creating new hypotheses. Well, that's usually the way research is working these days. Now, deciding the right approach for you, I wouldn't know at this point where to start. I don't know your research projects. Yeah, you know your research projects. You know your research objectives. Yeah, so deciding where to start is usually the trickiest part. Let's just move on to your objective. How it has to be formulated, why you formulate it like that. It has to be clear. It has to be something that everybody understands what you're trying to do. It has to be measurable. That means you will get some data that actually relates directly to what you wanted to obtain. It could be qualitative or quantitative data. It could be phenotypic or genotypic data. It could be scores. It could be alignment. It could be anything, but it has to be measurable somehow. If it's not measurable, then you're just using rhetoric. You're just saying nice things about grandiose objectives of what you want to get, yeah, but you will not be able to actually say, oh yes, I finished, because you cannot measure how you finished. If you haven't decided, if you haven't chosen an objective that can be measured, then you will never finish your objective. You will never reach your objective. It has to be as simple as possible, and this goes to the next one. Yeah, if your objective is too complex, or it can be decomposed into smaller objectives that are more clear, more simple, do it. Do it. Decompose them. You can put as an overall objective your big one, yeah, but try to understand that for your research, for your team, yeah, you have to obtain these minor objectives first. The same as before, yeah. If you want to find correlations between genes and a specific phenotype, go step by step. It is a good objective, a good overall objective of a big project, yeah, but it has a lot of small intermediate steps. And each of those steps will have its own problems, its own hypothesis, its own way of solving. And each of those steps will depend on what are your capabilities, skills, and the facilities that you have available for yourself. So do it in a smaller objective. That's going to make things simpler for you. That's going to let you know that you are moving forward in your research and that you're not stuck in some part because you haven't achieved your big objective yet, but you do have a smaller objectives that you've been accomplishing slowly but surely. And finally, just be realistic. Just be realistic about what you expect and what you can get. This actually goes in several ways. First, if you're not a NASA scientist or if you don't have anybody known from NASA, then you probably won't be able to know how potatoes grow in Mars. That's just not going to happen. Yeah? Or how a bacteria that was taken to the moon has changed its genomic background or genomic methylation to get used to being in this foreign environment. That's not going to happen unless you do have it. So be real. You do have those contacts. So be realistic. Also, this also goes to if you're not familiar with a method that you do know you will need, realistic that you don't know all the parameters that are, or all the details of that objective, or of that method. Don't be ashamed of asking questions, going to the right person or asking around if somebody else knows the right question. Even if you go to Wikipedia to just start getting to understand something, nobody knows everything. So just be realistic about what you know and what you don't know. So I have a few examples here. So let's see the first one. The first one, it says, my project X or project 1. I want to discover why people prefer Coca-Cola over Pepsi. Is it clear? Do you think it's clear? Yeah, they want to know why people prefer Coca-Cola over Pepsi. Yeah, it's very clear. Now, does it give you an actual measurable objective? Can you measure something that will tell you, oh yes, this is the reason why Coca-Cola, from all these reasons, this is the reason why people prefer Coca-Cola over Pepsi? Does it give you something as clear as that? Even though it looks like, hey, yeah, it sounds like, it sounds feasible, yeah? Do you have anything measurable there? What, there is something measurable there? That is a good point. How would you reformulate this one so that you include that one? Would you put just discover why people prefer Coca-Cola, or would you use something that, oh, my hypothesis or my objective is to demonstrate that Coca-Cola generates more serotonin than Pepsi, and therefore people like it more? Which one do you think it's easier to measure? It's more, not only narrow, but it's more to the point. The one you're saying, that's a lot better than what I put there. If you just want to know why people prefer Coca-Cola, that's just a random question. Like, why do people prefer the number 1 over number 2? I don't know, people like it. But yeah, maybe it has something to do with the sound of the number 2 over the number 1, and it elicits some hormones in your brain, and it makes you feel better. Why do people prefer opera over rock and roll? Or maybe the other way around. Why do people prefer chocolate over vanilla? But that's the thing that you pointed out, that you're trying to look for something to measure. For example, the amount of serotonin produced by one treatment or the other. Yeah, that's a way to measure it. This one doesn't give you that clue. This project 1 doesn't give you, hey, what am I going to measure? Yeah, but that's exactly what the question is aiming for. It's like they don't know the factors. So, yeah, so would you like, if you were reviewing a project and you get something like, would you say it's something feasible to do? Just from that look. You may have your own ideas of how to measure it, yeah, but has the scientists, the researchers, been clear enough about how he's going to do it? In only this? I like your opinion. It could be serotonin, it could be something else, right? But you already had an hypothesis. Yeah, it is serotonin that causes people to like this more. Yeah, that is his hypothesis, and that's something that can be measured. Yeah, this thing here, it sounds nice, but it doesn't really give you any clues of how they're going to tackle or how they're going to approach this. Number 2, identify factors related with pet aggressiveness. That is my objective. I want to identify factors that correlate to pet aggressiveness. Yeah, first, if you're saying pet aggressiveness, how do you measure pet aggressiveness? How do you know that some pets are more aggressive than others? We can feel it, but can we measure it? How do we know that some pets are more aggressive than others? Because if we don't measure that, how can we correlate that aggressiveness, that different measure of aggressiveness, to any factor that we want to test? We could test serotonin. We could test diet. We could test, I don't know, some genes. We could test a lot of things. Maybe psychological things. We can try to correlate it to psychological background. Maybe they were abandoned by their mother when they were just still lactating. Maybe they were split from their mother when they were still lactating, and that's why my pet is more aggressive. There are a lot of factors we can correlate to this. How do we measure pet aggressiveness? Is that clear here? Is there a way that needs to be set? Project 3, determine the minimum temperature. Oh, sorry, this is autocorrect. The minimum temperature required to induce dormancy in seeds, not cells, seeds. How do you like that one? What do you think is missing, or you don't think it's missing anything? Just think about all the things. Can you measure temperature? Can you determine whether something is dormant or not? Can you correlate those? If you can do both, and if you can do the three of them, so you can determine the temperature, you can measure the temperature and manipulate the temperature, you can determine whether a seed is dormant or not, then you can determine can you determine the correlations? Yes, you can determine the correlations. So can you actually tackle this objective? What do you think? Yeah, yeah, that's true. That's true. You will have to say, yeah, what kind of plant, right? What species, or maybe even what variety. No, different varieties will probably have different dormant temperatures. Yeah, but overall, the fact that you're trying to correlate temperature to dormancy, it's probably measurable in every species. You would have to be more concise about, yes, this specific species, because I'm not going to use all the plants in the world, right? But even whatever species I choose, I can measure all those things. Provided, that's another thing, yeah, provided it goes into dormancy. Maybe that's it. There is no minimum temperature. Now, finally, number 4. Measure the heavy metal sequestration rate of three sugarcane varieties, because I want to use them for bioremediation, for example. Can we measure heavy metal sequestration rate? Or I don't know if that's the right word. I'm not too familiar with bioremediation. I just put it there randomly. But the idea is capture, like they absorb it from the environment. Can we measure that? If we can't measure that, then we can compare the 3 varieties based only on that measure. Just measure. And the objective is very clear. I'm just going to measure. I'm not going to draw any conclusions out of it. I'm not going to do any correlation between it. I'm not going to do any recommendations from it. I just want to measure. This is probably just a sub-objective or a minor objective of a bigger research project. But it is clear and it is achievable. It's not what we call a moving target. If you ask too broad a question, it's usually a moving target. The same thing goes for data exploration alone. Data exploration alone is like you can explore forever. Forever. I was given the example yesterday of, let's assume you're in the middle of the Atlantic Ocean or the middle of the ocean, yeah? And you start moving around. You want to go to your land, to land. You want to see your family again. So you can start swimming in one direction. You can either choose to start moving around, trying to see, maybe I can see some land from here. Oh, I didn't see anything. Maybe from there, maybe from there, maybe from another part, maybe from another part. Or you can just decide, okay, fuck it, I'll go that way, and I'm not gonna stop until I see something. You can be doomed. Yes, you can just never reach home. You may drown. Yeah, you may drown. Yeah. But you went in that direction, and you got, in the end, you got information. What? This wasn't the right direction. You should have gone in a different direction. But if you start splashing around the middle of the ocean, you will also drown. You will never get anywhere. Now, your background research. This is important. We were discussing this at the beginning of the presentation. Basically, why are you doing your research? Your background research, your literature research. What is the main objective of doing your literature research? Remember, it's not only to fill in a form. It's not only because they asked me to do background research in order to fill a grant application form. That's not the only reason. Because way too often, I see a lot of people who just cram a lot of information, some of it related, some of it not related to what their objective is. But if you're focused, yeah, you will be able to determine which is relevant to your project and which isn't, because this is what you're looking for. First, you're looking for gaps in knowledge, gaps in literature, things that nobody has thought or nobody has published yet, but that you have that idea already. You want to tackle that. Yeah, you want to address something that hasn't been addressed yet. Second, you don't want to repeat other people's experiments, at least without adding something on your own twist. Yeah, because it's futile. It's not going to be publishable. Yeah, you're probably going to be struggling with trying to repeat exactly the same conditions that the other researcher tried to do. Yeah, you will be better off just going as a visitor to that lab that you're interested in, or to that, or just communicate with the researcher that has already done this, and asking, hey, how did you do it? What were the caveats? What would you recommend? You would be a lot better off. Also, why? You need to know what resources are already available. For everything you do, you need to know what's already there. If you don't know what's already there, you don't know where to start searching. If you're planning to do SNP discovery, if you're planning to do a microsatellite mining, if you're trying to do a correlationship between genes, etc., you need to know, hey, do I have genomic resources available for this? Do I have protein interaction resources available already? Do I have Do I have functional annotation of the proteins of a close relative of my plant? Does any plant closely related to the one I'm trying to study has any resource? If mine doesn't, does any plant that is close to the one I'm trying to work with has these resources already developed? If not, do I have the resources to develop this data before start as a first step towards my bigger goal? And that goes to the last point. You may need to develop the missing resources to help achieve your own objective. For most of us studying things from South America, plants, crops, animals, bacteria, etc., that's very often the case. Very little has been done in terms to produce the resources. And when the resources have been produced, very often they are hidden. Nobody knows about them, or very few people. Only people related to that project that generated the resources knows about it. Now, here we're going to talk. I cannot go through every single method that you will find to tackle each single step of your research. I don't have 7 years to finish this thing. So let's just think about how you select your approach. All right, these are just general recommendations. We can, and hopefully we will, see some of your own presentations today, to see what you're trying to achieve, how bioinformatics fits into it, what approaches you're proposing to tackle your objective, and maybe somebody here will give you a better idea because they have already seen something similar, or because they have already, they have read something about it, and they can give you some recommendations of how to design or improve your experimental design. So first, based on your objective, determine what kind of data you need to collect and what kind of a statistical analysis, if any. So this is extremely important. We talked about how the objectives need to refer to something measurable. So if they are related to something measurable, you need to know what kind of data you're going to get. This is the most crucial part. Understand the kind of data you're going to get. It's just as simple as saying, oh, I want to calculate the weight of something, but I don't understand what are the units I am using. If I'm using grams, if I'm using kilograms, or if I'm confusing density, grams per square meter with actual weight, or if I'm confusing distance or height with other variables. If I don't have a clear understanding of what is the data I'm measuring, then I need to stop. I need to open Wikipedia. I need to talk to an expert to tell me, hey, what exactly am I measuring here? So then, after you have a clear understanding, or at least a good understanding of what data you're going to collect, you're going to get, then you browse through all the methods that will produce that data. Let's say that your data needs to be sequenced, just for the sake of it. Just bear with me for a second. It's sequenced. How many sequencing methods do we have? Does anybody have any idea how many sequencing methods there are? Just to start with sequencing platforms. We have Illumina that has like 4 different machines. Sanger, which has like 3, I think, 2 or 3 different machines as well. Oxford Nanopore as well, which has like 4, if I remember correctly, 4 different machines as well. Pacific Biosciences, which currently has only 1, which is the sequel. Well, that's all I can think of from the top of my head. I know there are some Illumina-based sequencing, new sequencers, but they are not as popular. You used to have also something like Solid. You used to have something like 454. You used to have something like Ion Torrent. All these were sequencing technologies that they just fell out of use. They are no longer supported. They are no longer there. But the technology has already been developed. So it wouldn't be strange if for some coincidence or for some reason, somebody improves the technology, those technologies, and they become fashionable once again, because they offer better throughput, or because they increase their they increase the quality of the data they offer, etc. There are hundreds of reasons. Somebody can just step in and say, yeah, I'm going to fix this. But for now on, that's it. That's what you've got, right? And do all sequencers have the same features? have the same specs. Once you get the data, the raw data, your reads, do all the reads from all machines have the same error patterns? Do they have bias? Do each of them have different kind of bias? Like a GC bias, or maybe just related to a library construction bias, sampling bias, all this kind of. How does that affect your final, your objective, what you want to measure? You have to be very careful about that before actually saying, yeah, I'm going to sequence in Illumina, I'm going to sequence per end, I'm going to sequence 150 base pairs. That's it. Let's do it, because that's what everybody else is doing. Yeah, that does actually help your research, or doesn't, or it doesn't. Can you even build with your DNA extraction method? Can you even build Illumina libraries, or PacBio libraries, or nanopore libraries? That's the kind of things you need to consider. So, just this is no rule, because there are way too many methods. Minimum sample size, and this also goes for statistical analysis. If you're going to do a statistical analysis after that, yeah, you need to consider what parameters are the minimum necessary to have a successful or an informative minimum sample size, maximum resolution of the machines, or of the machines that you're going to use, minimum number of data points that you will need to actually find the correlation, or to actually find a significant value. You need to make sure that your results are reproducible. Yeah, reproducible. You need to check if the data is feasible to be collected. That is samples, FASTQ. Then you need to make sure that you have the appropriate infrastructure to not only collect, but maintain the data and analyze the data. You need to be aware of your time constraints. Some things are going to take a lot longer than you have, or some things are going to take a lot more money than what you've got available. So all these things are things that you need to be aware of when deciding what method or approach you're going to tackle. But the most important ones are, am I going to get the data that I need? Am I going to get the data that I need? All the rest can be fixed, or most of it can be fixed. If you can get the data that you need, yeah, you can just hire somebody else, or hire a better equipment, or better labs to actually run the tests that you wanted, or to run the analysis that you wanted, yeah, if it's in your budget. But if you don't have the right data, it doesn't matter how much extra money you put into trying to analyze it, you won't get the results you wanted. You won't be able to test what you wanted to test. Finally, limitations and caveats. This goes back to the method that you used. Yeah, each methodology presents its own limitation. It will have its own pros and cons, and that will limit the kind of conclusions to draw from the results. You can use two different machines to measure the amount of chlorophyll in a leaf. You can use two different machines, or you can use three. Which one has better resolution? Which one has better throughput? That's the kind of things that you need to think before you actually draw conclusions from the data you get. Also, be aware of too good to be true results. If something is too good to be true, it probably isn't true. It probably is an artifact, or there's probably a bias in your data. So be very aware. I always distrust any correlation or linear correlation data that shows me 0.99 R squared. If you show me 0.99 R squared in the data, either there is some bias in your data, or you fix your data somehow to actually make it match like that. Very few things well, at least in biology, they'll have a perfect. We all know that. We've been in the field, we've run experiments, and the most likely scenario is that usually we see no correlation in between what we were expecting and what we're getting. That's usually the case. We need to clean up the data and remove some data from the data to remove some data points from the data, because we know that, oh no, that plant died in the day 4 and didn't reach day 20, therefore it doesn't make sense to include it in the final result. All these kind of things, yeah, we've all done it, or we've been instructed to do it by our supervisors. Yeah, that doesn't make sense. Remove that. That comes with experience, yeah. But that's why I say don't trust too good to be true results. Your study will also be limited by the species you use, the environmental conditions you're working on, the geographic locations you're working on, a specific strain or variety of the plant species, which goes to what Andrea was saying before. Like, you need to be clear about what species you're working with. You need to be clear about where you're working with, where you're working these things. In plant growth, things as simple as atmospheric pressure will have an effect on the plant growth. Yeah, and sometimes we just cannot handle it. We just cannot manage that. That's why we have different results between cultivating in the coast and cultivating in the Andes, or cultivating even the amount of humidity in the atmosphere, cultivating in the jungle. Yeah, all these things, sometimes we cannot control them. So we need to be aware of what we're controlling and what we're not controlling, because that's going to have an effect on the conclusions we draw. Finally, the statistical tests have limits. If your data does not meet the conditions for a test, don't use that test. A very clear example I've seen way too often, yeah, is, I don't know, have you ever used a program called Structure? It's to determine population structure from just raw SNP data or microcellular marker data. Basically, what it does, it asks you to feed it number of different populations that you're putting, the classification of each of the samples that you're feeding it as belonging to population 1, 2, 3, or 4, yeah, and then it will try to find a certain frequency of markers that are more predominant in one population than in the other. If such a structure of the markers of the alleles exists in your data and they are clearly correlated with your data, then you say, yeah, there is population structure, which means that the populations are far enough that there is no DNA, there is no reproduction happening or gene flow happening between the populations, or it's very limited, right? That's basically what the program structure does. That's what it tells you. But when you look at the statistics of the test that it's running internally, it actually is, it's measuring that by the level of heterozygosity in the sample. So if it's measuring heterozygosity in the sample, why would anybody would try to use the structure software for mitochondrial genome, for chloroplast genome, or for bacteria? Can these mitochondrial, chloroplast, or bacteria, can any of these be heterozygous? They can't. They are haploid. They don't have two copies of it, or two different copies, one coming from the father and one coming from the mother. They have one single genotype in the species. But a lot of people use it. You will see papers that use the software structure to actually predict population structure using mitochondrial data. That's simply the wrong approach. That's not the right test for the data you're trying to analyze. So your statistical tests, again, have limits. If your data does not meet those conditions, don't do it. The same goes for GWAS. GWAS has a very specific set of rules. You have different kind of GWAS, that's true. And they will try to fit your data, or they will try to fit to the data you have. But if you have certain kind of data and you choose the wrong GWAS test, your results are not going to work. You need to talk to specialists in GWAS. Now let's talk about some overall methods. If you have any questions, this is just going to be very fast. This is going to be very fast. So, methods for sequencing. We were just talking about that. When are they useful? They're useful when you're trying to discover genotypes or develop markers. When it's not useful, or when it's not enough, when you try to find correlations between phenotypes and genotypes, it's not going to be enough. You need more data. Yeah. What kinds of sequencing you have? You have whole genome sequencing, transcriptome sequencing. You have targeted sequencing. Yeah, and I'm skipping a lot more. Yeah. Do you have any questions about this? Oh, by the way, do you have any questions about anything I've already said this far? Or comments, or just tell me you're wrong, absolutely wrong, you don't know what you're talking about? All right, so you have whole genome sequencing, transcriptome sequencing, targeted sequencing. Just a few more methods. Genotyping by sequencing. This is another kind of targeted sequencing, if you wish. Yeah. When it's useful when trying to identify high density markers like SNPs. It's useful when you're trying to do some phylogenetics analysis, population structure analysis, GWAS and genetic mapping. These are useful, yes. Yeah, but for all of them, you usually need phenotypic data. If you don't have phenotypic data, GBS alone, it's not going to be enough. That's why it's also not sufficient when you're trying to find cause-effect relationships. You may even find relationships between the SNPs, but there is not going to be a cause-effect relationship. The two main types are going to be DD-RADSEQ, which is double-digested restriction sequencing, and SCIM-GBS. The SCIM-GBS is kind of like a whole genome shotgun sequencing, but in a very low depth, like aiming for between 2 and 10x coverage of your target genome, instead of aiming for 60 or 50 or 60x coverage. We can talk about that later as well. Even more methods, metagenomics. Yeah, they are useful when trying to determine community composition or functional composition. Yeah, but you have to aware that there are different ways to go about it. It's overkill when you're trying to just single out one single family in your community, one single species in your community, or one single function in your community. It would be overkill. You don't want to spend a lot of money just trying to find something very specific from your sample. The kinds, the most popular ones are ribosomal RNA sequencing, targeted for bacteria in 16S, eukaryotes in 18S, or bacteria again in ITS. Whole metagenome sequencing. Yeah, this is when you want to assemble genomes from your metagenomic sample. Yeah, you have to be very careful. You need a lot of data for this. Yeah, do you have the money? Does it actually answer what you want to achieve? Maybe it doesn't. So be aware of that. Protein enrichment, protein family enrichment and sequencing. Basically, well, I've seen this happening before. Some scientists are not interested in the diversity, the community of diversity, but the community of a specific protein family. So they just go to NCBI, they align a bunch of proteins that are from the same family, they discover some conserved motifs, they design degenerate primers to tackle those conserved motifs, and they amplify that on their communities, on their soil sample, on their environmental sample, and they sequence that. What you're going to get, it's not going to be 16S, 18S, or ITS. You're going to get all the orthologous or paralogous genes that are present in the community. Which of these genes are more prevalent? You'll be able to know which of these genes are more prevalent in your community than not. So even though the approach may be similar to ribosomal RNA sequencing, yeah, it's going to have its own set of caveats as well. Mispriming, misalignment. It's gonna find you non-functional proteins or non-functional genes, pseudogenes, etc. So you have to be careful about that. Just a few more. Bisulfite sequencing. If you want to know the methylation status of your DNA, this is useful when you're just trying to study methylation patterns in the genome. Yeah, it's not useful or it's not enough when you're trying to correlate methylation patterns to expression patterns, or to what types there are at the moment. You have whole genome bisulfite sequencing. This usually is targeted to when you want to understand. This requires a little bit more background, but basically non-CG or non-CHG methylation patterns. Reduced representation sequencing, it's the second one. It's when you're trying to tackle only CG-rich areas, the methylation level of CG-rich areas. Chromatin immunoprecipitation, it's when you're trying to determine methylation of histones and their relationship with methylation of DNA. Yeah, so all of these have different pros, different cons, and they provide different information. If that's not enough yet, you have IsoSeq, you have nanopore, you have linked-rich, you have optical maps. They are all different technologies. Yeah, these are not all sequencing. The first two are sequencing. Hi-C, it's kind of sequencing, but actually it offers you more information about links of the genome, different interactions of the genome between parts of the genome, different parts of the chromosome. Linked reads, they give you long distance information about intramolecular relationships. Also, optical maps, it doesn't give you sequence. It gives you patterns of restriction in long molecules. Variation or genome graphs, this is just computer science stuff. Basically, how to represent or how to explore diversity of the data in the most efficient way possible. How to use that to actually determine haplotypes or genotypes from your data. Synteny maps, what are the relationship between conserved in order between different species? Genetic maps, what is the relationship between different markers in a family? How do they segregate, and can I relate that to a specific phenotype? And on it goes. This is just an example of all the recent sequencing technology that we have developed in the last years, and their capabilities, their throughput. I don't know if there's a cost. Well, the time that it takes to produce data, etc. Yep, that's it from me at the moment. Do you have any questions, guys? So is there a coffee break at the moment? Let's see it later. Yes? 11. All right, yeah, we've got time. So would anybody like to use this time to share their project? Yes, you would? Yeah, awesome. Oh, no. You're going to ask a question. Yeah, go ahead. Yes. Basically, because when you try to find any kind of correlation, you need two sets of data points, or at least treatments. For example, if you just sequence, yeah, let's assume, let's think of an example. Can you give me one example at the moment from your own research? Okay. Let's think of an example. You're trying to isolate genes related to, I don't know, yeah, a stress response. Yeah, something like that. Yeah, it's very broad. It's genes related to stress response. Okay, let's say that you do transcriptome sequence, RNA-seq. Yeah, because you want to see if there is different expression between two treatments, right? So you choose treatment 1, which is non-stress. You choose treatment 2, which is stress. Yeah, you choose the same plant so that you avoid any kind of genomic background interference in RNA-seq expression. Yeah, and then you run the experiment. You take your samples at the time point, or at the point in the timeline that you consider is the most appropriate. Yeah, you take the samples, you send them to sequence, and you get the sequence. If you didn't get the phenotypes, or if you didn't record which plants were under this or that stress conditions, if you didn't record what was the response of the plant, the phenotypic response of the plant to this or that stress condition, can you correlate your expression levels to that? See? So just sequencing, it's not enough to actually inform you whether this had an effect on the phenotype you're observing or on the response you're observing. You need to measure that response. It really depends on the amount of data you're working with. It's, if you're working with bacteria, usually a laptop should be enough.
But the whole sequencing?
But if you have metagenome sequencing,
yes.
Yeah, it's not one bacteria, it's hundreds of bacteria. Yeah. Usually you would go for, it really depends on how much data you actually have. That's what really comes down to. Now, visualization, very often you just need a laptop. Because visualization, you do it from already analyzed data. So you finally got some tables, yeah, that show you some either statistics or some variables that have already been measured, and you have, it's like after processing all your data and processing your reads and getting the relationship, etc., you produce final tables. Yeah, even if they are huge tables, a few gigs in size, yeah, you can still visualize it in a laptop. Most laptops will have more than 4 gigs of RAM. Currently, they're coming in 8 or 16 gigs of RAM as a regular thing. So a laptop should be enough to render the data, the visual data. Yep.
But if I want to see all the transcription profiles between my DNA assemble, it's enough like my laptop for that?
Yep.
Okay. Please,
how does that, basically he's asking, how does that help understand your results, just visualizing, for example, sequencing depth or something like that? How does that help, or how does that inform of your final objective. Does that really help? That's something you need to ask very often. Is like how the graphs that I'm showing actually make my point clearer to the reader. You know?
Okay, if for example, if I want to know of a chromosome for a specific bacteria, what region is the most expressed?
Yep.
Can I graph that?
Yeah, there are several ways to do it, but then again, it's like if you have that region as most expressed, which is unlikely that you'll find just one region, because there are, in the bacteria you will have like things that are going to be expressed all the time, regardless of the treatment, regardless of what it is. Yeah, everything that has to do with metabolism, energy production, breathing, or fermentation, etc., that's always going to be active. Yeah, so the fact that, and that's probably going to have a lot more activity, ribosomal RNA production, etc., that's going to have a lot more activity than a specific extra function that you'll find in your data. That's usually the case. So you will see expression data all over the chromosome. You won't see expression data just on one part. You may want to focus on just one part. Yes, that may be your focus. Yeah, but does that really help your point to just show that, hey, yes, I have these levels of expression here, if you don't compare it to anything else in the genome? So should I call names and ask you to present your data, your work, or do you want to offer yourself as well?